afternoon, teacher. We are team 2, formed by Antonio, Ver, Joanna, Maite, Melanie, Achari, and myself. Let's talk about the cultivation of the Demiscocus Denitrificans sustainable strategy to improve nitrogen cycle in the terrestrial ecosystem. Nitrogen is a vital element for life, as it is part of the building blocks of living things and of the nutrients in soil. However, excess nitrogen compounds such as nitrates can cause environmental problems. The bacterium Demiscocus denitrificans participates in the nitrification, a natural process that transforms nitrates into gaseous nitrogen, helping to maintain the balance of the ecosystem.
Okay, we're going to prepare a culture medium with nitrates to grow the bacterium Demiscocus denitrificans. We want to see how this bacterium transforms the nitrates into gases, which is an important part for the nitrogen cycle. Also, we are going to control the growth conditions such as temperature, light, and humidity. Ensure the bacteria develop properly. Finally, we want to relate the results for environmental sustainability by understanding how these microorganisms help to maintain the natural nitrogen balance for environment.
Materials: Demiscocus denitrificans, strain particle found on various surfaces, swabs, culture medium, nutrient agar with a nitrate solution, distilled water buffer solution to adjust the pH, preferably pH 7, gloves ground and other personnel, team, petri dishes, test tubes, pipettes or micropipettes, optical microscope, pH meter or pH reagent strips.
Preparation of the culture medium: dissolve the agar and the nutrient, including the nitrate source in water. Stated, adjust the pH to approximately 7 using the buffer solution. Sterilize the medium either by autoclave or following a method. Insurance available in the laboratory. 2. Inoculation. When the medium is cold, inoculate a small amount of the Demiscocus denitrificans in petri dishes and in test tubes. Label each sample to keep a record. Incubation. Keep in a place at room temperature for 48-72 hours. Observation and registry. Observe bacterial growth periodically with the microscopes. Record changes in the appearance of medium, such as modifications in the color that indicate the transformations of nitrates. Compare the results with a group control media with inoculation to confirm that the changes are due to the activity of the bacteria. Analysis and discussions. Analyze the information collected to determine the effectiveness of the bacteria in the reduction of nitrates. Relate the results to the importance of maintaining a balance. The nitrogen cycle, highlighting the potential of these bacteria to contribute to a more sustainable terrestrial ecosystems.
The ambient temperature favors the growth and metabolic activity of the bacteria, indicating that these conditions are suitable for the cultivation and denitrification function. Has resulted after 36 hours. We observe growth in the culture fungi at 3 small purple spots, which are our bacteria. Under a microscope, this is a tier.
Efficiency in the nitrification. The reduction of nitrates was most evident between 14:8 and 72 hours of incubation, suggesting that the Demiscocus denitrificans reaches its peak denitrification activity during this period under the experimental condition. Used. Optimal growth condition. Neutral pH and room temperature favor the growth and metabolic activity of the bacteria, indicating that these conditions are suitable for its cultivation and nitrification function.
Conclusion. This experiment demonstrates that the bacterium Demiscocus denitrificans transforms nitrates into gases, reducing their concentration, which is vital for the nitrogen cycle and preventing water pollution. Its agricultural and environmental uses proposed a sustainable strategy compared to chemical methods, research into its effectiveness in various soils and its potential large-scale effects is recommended.